The fast M&M batch correction is batch correction that was originally implemented in Python, but now via the integrate layers function in Seurat, you can also use it in R. So it will cluster all the cells together, so of all the samples in one common gene expression space. So you will get something like this, where you have cells from batch number 2 here and cells from batch number 1 here, but they are clustered in the same space. And then for every cell in this space, he's going to look for the nearest neighbors, so the cells that are the closest by, basically. So, and in this process, he will try to find nearest neighbors that are mutual for cells from different batches. So this is what you see here. This is a cell from batch 1. This is a cell from batch 2, and they both have the same nearest neighbor. This is a mutual nearest neighbor. And then he's going to use these mutual nearest neighbors to perform a correction. So to move the cells in the gene expression space towards each other. So, and this is what you see here. For instance, these two clusters have mutual nearest neighbors. So he's going to move them towards each other. And this is the end result. They are very close to each other. Also here, these two clusters have mutual nearest neighbors, so he's going to move them towards each other until they form one big cluster. If there are clusters that do not have mutual nearest neighbors with clusters from other batches, he's just going to leave them as they are. And this is what you see here for the pink cells. Here, the nearest neighbors of this cluster are the blue cells from the same batch. But since he's going to move the blue cells towards the blue cells of the other batch, he's going to move these yellow cells along with the blue cells. So they will also move and they will end up here, as you can see. And then you can repeat this process. So you start with 2 batches. If you have more than 2 batches, you first do it for 2 batches. You end up with this. And then you can add a 3rd batch if you want to. And you will repeat the process. So the stringency of the correction, and for this I am basing myself on publications, is somewhere similar to what Harmony is doing. But to be honest, inside the VIB, there are no people who are using this batch correction. So I don't have first-hand information about the stringency of this batch correction. But according to the benchmark studies, it kind of behaves similar with respect to stringency as Harmony. So you can use it when you have different cell types in the batches. You can see that here for the pink cells. So the pink cluster here is a cell type that only occurs in batch number 1 and not in batch number 2. But he will just leave it alone and leave it as it is, so he can perfectly handle this. And you use it when you have not that harsh batch effects. So typically here in this side of the figure here, so for removing batch effects between replicates or between patients.